Welcome to the Deep Dive, where we take your burning curiosity, our rigorous research, and a stack of expertly curated sources, and transform them into knowledge that truly helps you master even the most complex topics. Today, we're embarking on a very special deep dive into a subject that fundamentally impacts every single one of your estimated 40 trillion cells. We're talking about the intricate world of hormonal regulation of energy metabolism. You've provided us with incredibly rich source material, and our mission today is to truly unpack how your body constantly manages its fuel supply and demand, from digestion to fasting, and why getting this balance right is so crucial for your health. Our goal, to leave you feeling like an elite professional in this field with a deep understanding of every concept.
It's a fundamental biological challenge, isn't it? The human body needs a continuous energy supply for all cellular work, even though we only eat intermittently. Think about it. Your body actually produces the equivalent of its own body weight in ATP, that's adenosine triphosphate, the cell's energy currency, every single day. So we're going to explore precisely how different fuels are used and stored and how powerful hormones act as the ultimate conductors of this metabolic orchestra, making sure that energy is always available for processes as vital as maintaining cellular structure, synthesizing complex molecules, contracting muscles, and of course, thinking.
That's a staggering amount of energy production to manage. Okay, let's unpack this incredibly complex system. We'll break it down into digestible insights that will allow you to, truly master this information. So to kick things off, let's ground ourselves in the fundamental challenge. Why is this continuous energy supply such a big deal for our bodies? What's the core problem, that our metabolism has had to solve over evolutionary time?
You've hit on the central point. You have 40 trillion cells, and they are continually performing work. This ranges from Our diet provides three primary types of fuel. First, monosaccharides, which are simple sugars like glucose, fructose, and galactose. Second, long-chain free fatty acids, or FFAs. And third, amino acids, or MAs, the building blocks of proteins. The fourth general type of fuel is ketone bodies, or KBs. Now, these are largely absent in the diet. Instead, KBs are produced by the body, predominantly by the liver, during periods when other fuels, especially glucose, are scarce, like during fasting.
And when we talk about metabolic phases, it's clear the body doesn't handle fuel the same way right after a big meal as it does during an overnight fast. Can you walk us through those critical phases? Because they really dictate everything that follows, don't they?
Absolutely. There are three general metabolic phases, dictated by the abundance or scarcity of fuels, and they are exquisitely orchestrated by phase-specific hormones. First, we have the digestive or absorptive phase. This occurs during the 2 to 3 hours it takes to digest and absorb a meal. Then there's the interdigestive or post-absorptive phase, which normally occurs between meals. And finally, the fasting phase. Most commonly experienced overnight, say between your last snack and breakfast, prolonged fasting or starvation are simply more extreme forms of this. We also have to acknowledge physical exertion as another important metabolic phase, with its heightened energy demand. For our deep dive today, though, we'll primarily contrast metabolism between the digestive and fasting phases.
Beyond these phases, certain cell types really punch above their weight, having an outsized role in whole-body metabolism. Which are those key cellular powerhouses?
Well, while every cell is involved in energy metabolism to some degree, three cell types have a profound impact on whole-body metabolism. These are hepatocytes, your liver cells, adipocytes, your fat cells, and skeletal myocytes, your muscle cells. And it's crucial to understand that during the digestive phase, these three cell types often function largely independently. But in stark contrast, their actions become highly integrated and coordinated during the fasting phase, working together to maintain adequate blood glucose levels while simultaneously providing alternative energy substrates for each other.
And speaking of blood glucose, why is maintaining it within such a narrow range absolutely vital for our survival? It feels like the central nervous system's entire lifeline depends on it.
It truly is. Cells with few or no mitochondria, like red blood cells or the lens cells of the eye, are absolutely dependent on glucose for their energy. And critically, the central nervous system, or CNS, can only use glucose for ATP production under normal conditions. So during the interdigestive and fasting phases, maintaining blood glucose above a certain minimal threshold, for example, above 60 milligrams per deciliter is, non-negotiable to avoid CNS-related symptoms. If blood glucose falls too low, you'd first experience symptoms from a hypoglycemia-activated autonomic response, like nausea, sweating, or cardiac arrhythmias. If it continues to drop, it progresses to what's called neuroglycopenia, causing cognitive dysfunction, loss of coordinated motor function, and ultimately even coma and death.
That's the terrifying lower limit. But what about the upper limit for blood glucose? Because it sounds like your body is just as vigilant about keeping it from going too high, right?
Precisely. Glucose is a fairly reactive molecule. Chronically high blood glucose, even if not immediately life-threatening like severe hypoglycemia, leads to pathologically high intracellular glucose in many cells. This then causes non-enzymatic covalent linking of glucose to proteins and other molecules, disrupting their configuration, half-life, and function. This damaging process is collectively referred to as glucotoxicity.
And the implications of glucotoxicity are profound for long-term health, especially in the microvasculature.
Yeah.
That's where we see the real damage in conditions like poorly controlled diabetes.
Exactly. The endothelium of the microvasculature, specifically in the kidney and retina, as well as the ventral nervosum of the autonomic nervous system, are particularly sensitive to hyperglycemia. Glucotoxicity is, in fact, the root cause of the nephropathy, retinopathy, and peripheral neuropathy that occur in poorly controlled diabetes mellitus. This underscores why whole-body metabolism must minimize the magnitude and duration of any rise in blood glucose after a meal and maintain it below a safe maximal threshold, typically 100 milligrams per deciliter, during all other times. For context, fasting blood glucose between 100 and 124 mg/dL indicates impaired glucose tolerance, and values at 125 mg/dL and above are evidence of diabetes.
So considering these incredibly tight thresholds and the vast number of cells involved, how does the body orchestrate this delicate balance across such varied states and cell types? Sounds like a master plan.
This is where the hormonal orchestrators come in. Metabolism during the digestive phase is almost entirely driven by insulin. During the fasting phase, insulin levels drop, which is itself a key signal for metabolic adaptation. In addition, glucagon and catecholamines, epinephrine and norepinephrine become the primary stimulators of metabolic pathways, integrating the body's entire response to the absence of ingested fuels. Growth hormone and cortisol also contribute, though their roles are somewhat minor in this immediate context.
Okay, let's follow the journey from the beginning then. So you've just enjoyed a meal. What's happening inside your body in that digestive phase? And how does insulin, this master hormone, manage everything to prevent that glucose surge and efficiently store fuel?
Right. In the digestive phase, the core objectives are crystal clear and perfectly aligned with insulin's job description. First, prevent high blood glucose from damaging tissues. Second, synthesize and store fuel polymers like glycogen, triglycerides, and proteins. And third, promote overall anabolism, which is the building up of complex molecules from simpler ones. Insulin is the absolute director here. It ensures that the incoming glucose and other nutrients are either utilized immediately for energy or efficiently stored for later.
Okay, let's break down exactly how glucose gets into cells and then how it's processed. It's not just a free-for-all, is it? Not at all. Glucose first enters cells via facilitative GLUT transporters, like GLUT1, 2, 3, and 4. Once inside, it's critically trapped and activated by being converted into glucose 6-phosphate, or G6P, through the action of enzymes called hexokinases. Why is this trapping critical? Because G6P cannot pass back out through GLUT transporters, effectively locking it inside the cell. From G6P, cells can metabolize it to pyruvate via glycolysis, which produces a small amount of ATP without requiring mitochondria or oxygen. Cells without mitochondria, like red blood cells, simply ferment pyruvate to lactate and export it. Most other cells, however, import pyruvate into their mitochondria, convert it to acetyl-CoA by the pyruvate dehydrogenase complex, and then cycle it through the tricarboxylic acid, or TCA, cycle. This metabolism ultimately releases CO2 and generates large amounts of ATP through the electron transport system and oxidative phosphorylation, a process that absolutely depends on oxygen.
So insulin has these very specific, almost tailor-made actions depending on the tissue. How does it direct glucose in the liver, for example, given how central the liver is to overall metabolism?
Yes. Insulin's actions are remarkably tissue-dependent. In hepatocytes, your liver cells, glucose uptake occurs via GLUT2 transporters. Now, these are not directly regulated by insulin for their insertion into the cell membrane. However, insulin profoundly increases hepatic retention and utilization of glucose. How? By increasing the expression of an enzyme called glucokinase. Glucokinase is a low-affinity hexokinase that acts as the liver's primary glucose sensor, meaning its activity scales with glucose concentration. Insulin does this by increasing the expression and activation of the transcription factor SREBP1C, a master switch that coordinately increases several enzymes involved in glucose utilization and de novo lipogenesis, or DNL, the process of making new fat. And to prevent a wasteful futile cycle where glucose is made and immediately broken down, insulin also represses the gene expression of glucose 6-phosphatase, or G6-pase. That's the enzyme which would otherwise convert G6P back to glucose for export. It achieves this by phosphorylating and inactivating FAXO1, a transcription factor that stimulates G6-based gene expression.
So insulin is really pushing the liver to not only keep glucose inside, but also to actively use it. How does it then direct the liver to store that glucose as glycogen?
Good question. Insulin indirectly increases glycogen synthesis through that increased glucokinase expression we just discussed. High G6P allosterically, meaning by binding to a site other than the active site, increases glycogen synthase activity. More directly, through specific protein phosphatases, insulin promotes the dephosphorylation and activation of glycogen synthase, pushing glucose into storage. Simultaneously, insulin inhibits glycogen phosphorylase, preventing the breakdown of glycogen and thus blocking yet another futile cycle of synthesis and breakdown.
And what about glycolysis, the process of burning glucose for immediate energy? How does insulin boost that commitment reaction in the liver, really ensuring that glucose is funneled into these pathways?
Right. Insulin significantly increases glycolysis by activating phosphorfructokinase 1, or PFK1, which catalyzes the commitment reaction for glycolysis. That's the phosphorylation of fructose 6-phosphate to fructose 1-ferrocycle-6-bisphosphate. Insulin also cleverly inhibits the reverse reaction catalyzed by the gluconeogenic enzyme fructose 1-ferrocycle-6-bisphosphatase. It achieves this through a fascinating bifunctional enzyme called PKFBP. Insulin-activated protein phosphatases dephosphorylate PKFBP, which activates its PIK2 kinase function and simultaneously lessens its F2-ferrocycle-6-bisphosphatase function. This leads to higher levels of fructose 2-ferrocycle-6-bisphosphate, which then allosterically activates PFK1 and inhibits fructose 1-ferrocycle-bisphosphatase. Insulin also activates pyruvate kinase, or PK, which catalyzes the conversion of phosphoenolpyruvate to pyruvate by activating a protein phosphatase that dephosphorylates it, and it increases pyruvate kinase gene expression via SREBP1C. Finally, insulin activates the pyruvate dehydrogenase, or PDH complex, which converts pyruvate to acetyl-CoA for entry into the TCA cycle.
That's an incredible level of intricate control over glucose. But insulin also plays a huge role in fat synthesis in the liver, what you call de novo lipogenesis. How does it orchestrate that?
Absolutely. During the digestive phase, some of the acetyl-CoA produced from glucose metabolism is transferred from the mitochondria to the cytosol as citrate. This citrate is then converted back to acetyl-CoA by an enzyme called ATP citrate lyase. Insulin significantly increases the gene expression of ATP citrate lyase through SREBP1C. This cytosolic acetyl-CoA then enters the fatty acid synthesis pathway. The first committing step involved converting acetyl-CoA to malonyl-CoA by acetyl-CoA carboxylase. Insulin stimulates both its gene expression via SREP1C and also promotes its dephosphorylation, activating the enzyme. Malonyl-CoA is then used by fatty acid synthase, or FASN, to produce longer fatty acids. FASN gene expression is also dramatically enhanced by insulin through SREBP1C. The necessary reducing power for this process in the form of NADPH largely comes from the pentose phosphate pathway via glucose 6-phosphate dehydrogenase. And insulin also increases G6PD gene expression, again through SREBP1C.
And this is where the body gets really clever, preventing a wasteful cycle of fat synthesis and then immediate breakdown. How does insulin indirectly put the brakes on fat oxidation?
It's a brilliant design. By activating the steps that lead to the generation of malonyl-CoA, insulin indirectly inhibits the oxidation of FFAs. Malonyl-CoA inhibits CPTI, which is the critical transporter that moves FFAs from the cytosol into the mitochondria for beta oxidation or fat burning. So the FFAs that are synthesized by DNL cannot be transported into mitochondria to be immediately broken down. This increased malonyl-CoA effectively prevents that futile cycle of fat synthesis to fat oxidation. Beyond this, insulin also promotes the formation of triglycerides from these newly synthesized FFAs, and it acutely promotes the degradation of ApoB100, the VLDL apoprotein, preventing the liver from secreting VLDL during a meal when the blood is already rich with dietary fats in the form of chylomicrons.
Okay, shifting to skeletal muscle, what's insulin's biggest impact there, the one that makes the largest difference to how our body handles glucose overall?
In skeletal muscle, the most significant action of insulin for whole-body glucose tolerance is stimulating the translocation of pre-existing intracellular GLUT4 transporters to the cell membrane. This dramatically increases glucose uptake into muscle cells, representing the largest single impact on glucose clearance from the blood. Muscle also has a high-affinity hexokinase for trapping glucose as G6P, which, importantly, cannot be converted back to glucose and released into the blood because muscle does not express glucose 6-phosphatase. Insulin promotes glucose storage as glycogen in muscle and its oxidation through glycolysis and oxidative phosphorylation for ATP production.
And for adipocytes, our fat cells, how does insulin guide them during this phase?
Adipocytes also see increased glucose uptake via GLUT4 translocation. Insulin stimulates glycolysis in adipocytes, which is crucial because it generates glycerol 3-phosphate, or G3P, required for reesterifying FFAs into triglycerides for storage. Insulin also stimulates the synthesis and secretion of lipoprotein lipase, or LPL, into the subendothelial space of capillaries. This allows LPL to extract FFAs from dietary chylomicrons in capillary beds. It promotes the esterification of these FFAs into storage triglycerides. Finally, insulin directly inhibits hormone-sensitive lipase, or HSL, within adipocytes, thereby promoting the storage of FFAs as opposed to their release.
Insulin is clearly anabolic across the board, focusing on building and storing. What about its role in amino acid and protein metabolism?
Insulin consistently drives anabolic pathways for regenerating degraded molecules, including other amino acids, proteins, and nucleotides. In the liver, while amino acids are used for ATP synthesis, insulin primarily promotes overall protein synthesis and attenuates the activity of urea cycle enzymes, which normally break down amino acids. So it reduces overall amino acid catabolism.
You also mentioned chylomicrons, which carry dietary fats. How does insulin orchestrate their journey after a meal?
Right. Chylomicrons are formed in intestinal enterocytes from digested dietary triglycerides, along with other lipids, and the protein ApoB48. They're secreted into the lymphatics and then enter the blood circulation. While circulating, they acquire ApoE and ApoC2 from HDL particles, becoming mature chylomicrons. When they enter the capillaries of adipose tissue and also cardiac muscle, lipoprotein lipase, or LPL, which, as we discussed, is stimulated by insulin digests the triglycerides, releasing FFAs. Cardiac muscle preferentially uses FFAs for energy and extracts most of its FFAs from lipoproteins. LPL activity in skeletal muscle is relatively low in this fed phase. After digestion, the chylomicrons become smaller, denser chylomicron remnants, or CRs. These CRs are atherogenic, meaning they can contribute to plaque formation in blood vessels. Because they still contain ApoE, they are efficiently cleared from the circulation by the liver, which endocytoses them, and any remaining FFAs are reesterified into intrahepatic triglycerides.
So if I'm understanding this incredible symphony correctly, insulin acts like a master conductor, making sure all the newly arrived fuel is either used for immediate energy or efficiently stored away. Ready for when the food supply runs out, it's a total command. Now let's flip the switch. Imagine you're asleep or you've skipped a meal, maybe even for a few days. Your body shifts into the fasting phase. The objectives definitely reverse here, don't they? How do the counter-regulatory hormones then take over?
Yes, the objectives completely reverse. It's a metabolic pivot. The primary goals of the fasting phase are, first, glucose production to maintain blood levels above that crucial lower limit for the brain. Second, glucose sparing, meaning most cells switch away from glucose and begin to use alternative fuels. And third, overall catabolism, the breaking down of stored polymers to release those necessary fuels. In this phase, insulin levels drop to low levels, which is itself a key signal for metabolic adaptation. Then glucagon and catecholamines become the key players, along with vital intracellular metabolic signals.
The liver is clearly central to glucose production during fasting. How does it manage that, given that we're no longer eating carbohydrates, it's generating new glucose, isn't it?
Exactly. Hepatic glucose production, or HGP, relies on two critical metabolic pathways within the liver. The first is the rapid breakdown of stored glycogen through glycogenolysis. Hepatocytes express glucose 6-phosphatase, or G6-pisase, which allows them to convert G6P back to free glucose. This glucose can then exit the cell via GLUT2 to enter the bloodstream. However, liver glycogen supply is relatively limited and typically becomes exhausted in about 8 hours. The second and more sustained pathway is gluconeogenesis, which is the new synthesis of glucose from non-carbohydrate precursors.
And what exactly are those precursors for gluconeogenesis? Where do they come from when you're not eating carbs?
The primary precursors are lactate, glycerol released from adipocyte lipolysis, and gluconeogenic amino acids released from muscle proteolysis. These precursors circulate to the liver, where they are avidly taken up and used for gluconeogenesis. The regulation of gluconeogenesis is also critical. FOXO1, which was inactivated by insulin in the fed state, becomes highly active in the fasting state. It stimulates the gene expression of both G6-pase and PAPase, or phosphatidylpyruvate carboxykinase, both key gluconeogenic enzymes. Glucagon and catecholamines, via the PKA-CEREB signaling pathway, also significantly increase PPC expression. This reciprocal regulation with insulin is key. Insulin inactivates FOXO1, while its absence in the fasting state allows FOXO1 to activate genes for glucose production.
So the liver is diligently producing glucose, but most of their cells need to conserve it for the brain. How does the body achieve this glucose sparing and switch to alternative fuels?
Glucose sparing involves a general decrease in glucose uptake by most cells, especially skeletal muscle, and a profound switch to alternative fuels. In skeletal muscle and adipocytes, glucose uptake is greatly reduced because the insulin-dependent GLUT4 transporters move away from the cell membrane, existing in an inactive intracellular state. This means alternative fuels, primarily FFAs and ketone bodies, need to be delivered to these tissues to meet their energy needs.
And those alternative fuels, FFAs and ketone bodies, how are they produced and then utilized by the cells that need them?
FFAs are primarily released from adipocytes via lipolysis, where hormone-sensitive lipase, HSL, is activated by glucagon and catecholamines. FFAs are also released from intrahepatic triglycerides that were packaged into VLDL. These FFAs then undergo beta-oxidation to acetyl-CoA in various tissues. Ketone bodies, or KBs, are produced in hepatocytes via ketogenesis from acetyl-CoA, which largely originates from the breakdown of FFAs and ketogenic amino acids. KBs can then be used by non-hepatic cells, like the brain after several days of fasting, and especially skeletal muscle, for ATP production via a process called ketolysis.
Now, this is truly fascinating. The very cells that produce ketone bodies, the hepatocytes, don't actually use them. Why is that? Why doesn't the liver just keep them?
That's a crucial piece of the puzzle, and it highlights the body's remarkable efficiency in fuel partitioning. Hepatocytes only perform ketogenesis, not ketolysis. They lack the necessary enzyme, beta-ketoacyl-CoA transferase, required for ketolysis. This prevents a wasteful futile cycle within the liver itself, making KBs, only to break them down again, and ensures that these valuable ketone bodies are exported and made available for other tissues that can use them, particularly skeletal muscle, and after several days of fasting, the brain, which increasingly adapts to use KBs as a primary fuel source.
And during fasting, what happens to our body's protein? Is it sacrificed for fuel?
Yes, unfortunately. There's an overall catabolism of protein during fasting. We see net proteolysis, especially from muscle, to release amino acids for gluconeogenesis in the liver to maintain blood glucose. Similarly, triglycerides within adipocytes undergo significant lipolysis to release FFAs and glycerol, which also serve as precursors for glucose synthesis in the liver.
And what about VLDL metabolism during fasting? We talked about it in the fed state. But how does its role change when there's no food coming in?
During fasting, the liver becomes the primary source of circulating triglycerides. The intrahepatic triglycerides that accumulated from de novo lipogenesis and chylomicron remnants during the fed state are now packaged with ApoB100 protein and secreted as VLDL, or very low-density lipoprotein. F-oxyxO1, which is active in the fasting state, stimulates the expression of ApoB100 and other components necessary for VLDL assembly. Unlike the fed state, where LPL activity is low in epicytes due to low insulin, during fasting, cardiomyocytes and skeletal myocytes significantly express LPL. They avidly digest VLDL and providing FFAs to these muscle cell types for energy. This process generates intermediate density lipoprotein, or IDL. About half of this IDL returns to the liver for uptake and digestion. The other half undergoes further digestion by hepatic lipase, forming low-density lipoprotein, or LDL. LDL is triglyceride poor, but cholesterol rich, and notably is considered very atherogenic, especially in the face of endothelial damage. It's cleared by the LDL receptor, which recognizes only ApoB100. The liver is the major site of LDL uptake, clearing cholesterol for excretion as bile acids or storage as cholesterol esters.
We've now traced the journey of glucose and fats through these different metabolic phases, guided by hormones. But let's take an even closer look at how these key hormonal messengers are made, how they're released, and precisely how they signal to cells. Starting with the pancreas, the endocrine maestro.
Oh, okay. The pancreas contains the islets of Langerhans, which are the endocrine part of the organ, making up about 1% to 2% of its mass. These islets are composed of several cell types, primarily beta cells, which constitute about 3/4 of the cells and produce insulin located centrally within the islet. Alpha cells account for about 10% and secrete glucagon, typically located along the periphery. Interestingly, blood flows through the islets from the central beta cells to the peripheral alpha cells. This means that insulin, as it's secreted, can directly inhibit glucagon secretion from the alpha cells, providing a very elegant local control mechanism.
That's a fascinating micro-level control. Let's really drill down into insulin first, its structure, how it's synthesized, and precisely how glucose stimulates its secretion. This whole chain of events is critical.
Right. Insulin is a protein hormone related to insulin-like growth factors and relaxin. It's synthesized as preproinsulin, which is then converted to proinsulin in the endoplasmic reticulum. Proinsulin contains the amino acid sequence of mature insulin plus a C-peptide. In the Golgi apparatus, proinsulin is packaged into secretory granules, where proteases cleave out the C-peptide, generating mature insulin. Mature insulin consists of two chains, an alpha and a beta, connected by two disulfide bridges, with a third disulfide bridge within the alpha chain. It's stored as zinc-bound crystals and released by exocytosis upon stimulation, with equimolar amounts of mature insulin and C-peptide released. Insulin has a very short half-life, about 5 minutes, and is rapidly cleared primarily by insulin-degrading enzyme, or IDE, in the liver, kidney, and other tissues. Because it's secreted into the hepatic portal vein, about half of the insulin is degraded on its first pass through the liver before reaching the peripheral circulation. This means the liver is exposed to much higher insulin concentrations than peripheral tissues. Now, for glucose-stimulated insulin secretion, or GSIS, let's break down this precise chain reaction. Glucose enters beta cells via the GLUT transporter. Once inside, it's phosphorylated to G6P by glucokinase. Glucokinase is often called the glucose sensor because the rate of glucose entry correlates directly with its phosphorylation rate, which in turn correlates with the magnitude of insulin secretion. Metabolism of this G6P through glycolysis, the TCA cycle, and oxidative phosphorylation increases the intracellular ATP to ADP ratio. This crucial change then closes an ATP-sensitive potassium channel on the beta cell membrane, which depolarizes the cell membrane. This depolarization then opens voltage-gated calcium channels, and the increased intracellular calcium activates microtubule-mediated exocytosis of the insulin-containing secretory granules.
It's incredible how a simple rise in glucose initiates such a precise and orchestrated chain of events. And there's also the incretin effect, right? Which means ingested glucose has a greater effect on insulin secretion than glucose injected directly into the bloodstream. What's giving it that extra boost?
The incretin effect is due to the stimulation of incretin hormones from the gastrointestinal tract by ingested glucose. A clinically highly relevant incretin hormone is glucagon-like peptide 1, or GLP-1, released by L cells in the ileum in response to glucose. GLP-1 enters the circulation and binds to its Gs-coupled receptor on beta cells. This GLP-1 Rg-satinylase cyclase PKA signaling pathway amplifies the intracellular calcium effects, significantly boosting insulin secretion. Unfortunately, GLP-1 is rapidly degraded in the circulation by the enzyme dipeptidyl peptidase 4, or DPP4.
And this rapid degradation of GLP-1 has led to some really clever therapeutic strategies for diabetes, hasn't it? It's a great example of applying this deep knowledge.
Indeed. The understanding of this pathway has led to the development of several hypoglycemic drugs. Sulfonylurea drugs, for instance, directly close the ATP-sensitive potassium channels in beta cells. They effectively mimic the effect of glucose and stimulate insulin release. These are widely used oral hypoglycemics. Additionally, DPC4-resistant GLP-1 analogs and inhibitors of DPP4 are approved for type 2 diabetes, as they enhance the body's own incretin effect, leading to more robust glucose-dependent insulin secretion.
Beyond glucose and these incretins, are there other stimuli or inhibitors for insulin secretion? It seems like such a tightly regulated process.
Yes. Several amino acids and vagocholinergic innervation via muscarinic receptor 3 also stimulate insulin secretion by increasing intracellular calcium. Conversely, insulin secretion is primarily dampened by sympathetic autonomic regulation through alpha-2 adrenergic receptors. Binding of norepinephrine or epinephrine to these receptors decreases cyclic AMP, thereby dampening insulin secretion. This adrenergic inhibition serves to protect against hypoglycemia, especially during physical exertion, by reducing insulin release.
This level of detail in pancreatic function really brings to mind conditions like MODY, maturity-onset diabetes of the young. How do specific gene mutations tie into this intricate picture of insulin production?
MODY is indeed directly linked to gene mutations in pancreatic transcription factors, crucial for insulin gene expression and islet cell biogenesis. These include hepatocyte nuclear factor 4 alpha, HNF1, HNF alpha, insulin promoter factor 1, IPF1, and NeuroD1 beta 2. A heterozygous null mutation in any of these genes can lead to MODY. Similarly, mutations in glucokinase, that very glucose sensor we discussed, also give rise to MOY, highlighting its absolute critical role in beta cell function.
So once insulin is released and floating around, how does it actually signal to cells to tell them what to do? What's happening at the insulin receptor, and how does that communication system work inside the cell? The insulin receptor, or NSR, is a receptor tyrosine kinase, a type of receptor that directly phosphorylates proteins. It exists on the cell membrane as a homodimer. When insulin binds, it induces cross-phosphorylation of the receptor subunits on specific tyrosine residues. These phosphotyrosine residues then recruit and are bound by insulin receptor substrate, or IRS, proteins. The IRS proteins themselves are phosphorylated by the NSR, which then recruits phosphoinositide 3 kinase, or PI3K. PI3K converts a membrane lipid, PIP2, into PIP3. PIP3 is an informational lipid that recruits ACT protein kinase to the cell membrane where it becomes activated. This ACT kinase signaling pathway then orchestrates the numerous metabolic actions of insulin in hepatocytes, skeletal muscle, and adipocytes.
Okay, that's a lot of molecular signaling. So what does this all mean for the cell's metabolic activity? ACT sounds like a master controller involved in so many different actions. What are its key pleiotropic actions, as you call them, and how do they actually impact metabolism across the cell?
ACT's actions are indeed wide-ranging and truly at the heart of insulin's command. First, and critically, it directly causes the translocation of the GLUT4 glucose transporter to the cell membrane in skeletal myocytes and adipocytes, allowing a surge of glucose import. Second, it activates multiple protein phosphatases, which in turn regulate the activity of numerous metabolic enzymes in all insulin target cells, effectively turning various metabolic processes on or off. Third, ACT activates the protein complex mTORC1, which promotes protein synthesis and may inhibit proteaphosphol-mediated protein degradation, emphasizing insulin's anabolic role. Fourth, it activates the transcription factor SREBP1, especially SREP1C in the liver, which acts as a master switch orchestrating glycolysis and de novo lipogenesis for producing phospholipids, fatty acids, and triglycerides from excess glucose and fructose. ACT stimulates SREBP directly and indirectly through mTORC1, and SRBP1C also induces the enzyme G6PD in the pentose phosphate pathway, generating an ADPH, a required cofactor for DNL. And fifth, ACT inactivates the transcription factor FOXO1 by phosphorylating it, which promotes FOXO1's exclusion from the nucleus. This is crucial because, in the absence of insulin, FOXO1 would induce the expression of gluconeogenic enzymes and proteins involved in VLDL assembly. Beyond metabolism, the NSR also promotes proliferation and renewal of some target cells through the Resurov-MAPK pathway.
Now, let's shift our focus to glucagon. How is it structured and secreted, and what are its main signaling pathways, given its counter-regulatory role?
Glucagon, like insulin, is a peptide hormone. Its precursor, preproglucagon, actually harbors the amino acid sequences for glucagon, GLP-1, and GLP-2. In alpha cells, preproglucagon is specifically proteolytically cleaved to produce glucagon. Glucagon circulates unbound and has a very short half-life of about 6 minutes. The liver is its predominant degradation site, clearing as much as 80% of circulating glucagon in one pass. This means the liver is the primary target organ for glucagon, with lesser effects on adipocytes. Crucially, skeletal muscle does not express the glucagon receptor, which is an important distinction from insulin. The glucagon receptor is a G-linked G protein-coupled receptor, meaning its activation increases adenyl cyclase activity and thus intracellular CAMP levels. Glucagon exerts many rapid actions through pKa signaling and also has transcriptional effects by activating transcription factors like CRFM. The insulin-glucagon ratio is a major determinant of net metabolic effects. The primary stimulus for glucagon secretion is a decline in blood glucose. Insulin actually inhibits glucagon secretion, so low blood glucose has an indirect effect by removing that inhibition. Recent evidence also suggests low glucose has a direct stimulatory effect on alpha cells. Interestingly, serum amino acids also promote glucagon secretion, meaning a protein meal will increase both insulin and glucagon, which, as we noted earlier, protects against hypoglycemia, whereas a carbohydrate meal primarily stimulates only insulin.
And finally, how do catecholamines, epinephrine, and norepinephrine fit into this intricate hormonal dance, especially during stress or intense activity?
Catecholamines, epinephrine, and norepinephrine are other major counterregulatory factors. Epinephrine is primarily from the adrenal medulla, while norepinephrine is released from sympathetic nerve endings. They are released in response to decreased glucose, various forms of stress, and exercise. The CNS primarily senses hypoglycemia, initiating an integrated sympathetic response through the hypothalamus. Their direct metabolic actions are mediated primarily by alpha-1, beta-2, and beta-3 adrenergic receptors on muscle, adipose, and liver tissue. Like the glucagon receptor, beta-adrenergic receptors increase intracellular CAMP. Circulating catecholamines inhibit insulin secretion from beta cells via alpha-2 adrenergic receptors, while stimulating glucagon secretion from alpha cells via beta-2 adrenergic receptors, thus reinforcing the fasting state's metabolic profile.
We've now thoroughly traced the journey of glucose and its hormonal masters, as well as the flow of fats. But what about the broader picture of energy balance and appetite? How do fats travel through our body, and what other factors play a role in that bigger picture beyond just glucose?
This brings us to lipoproteins, which are essentially transport vehicles for lipids in the blood. The triglyceride-rich lipoproteins are chylomicrons and VLDL, or very low-density lipoproteins. They primarily deliver FFAs to muscle for energy and to adipocytes for storage. Cholesterol-rich lipoproteins include LDL, low-density lipoprotein, and HDL, high-density lipoprotein, which deliver cholesterol to various cells and remove excess cholesterol from the periphery. There are also the remnants of lipoproteins that have had their lipid cargo partially digested and are then cleared by the liver.
Let's quickly review chylomicron metabolism again in the digestive phase, and then move into VLDL in the fasting phase, clearly distinguishing their roles.
To quickly recap from the digestive phase, dietary triglycerides are processed in intestinal enterocytes into chylomicrons, which contain ApoB48. They enter the lymphatics and then the blood, acquiring ApoE and ApoC2 from HDL to become mature. In adipose tissue and cardiac muscle capillaries, lipoprotein lipase, or LPL, digest these chylomicrons, releasing FFAs. Cardiac muscle, in particular, extracts a lot of FFAs from chylomicrons for energy. LPL activity in skeletal muscle is low here. After digestion, chylomicrons become smaller, denser chylomicron remnants, or CRs, which are atherogenic. Because they still have ApoE, they bind to receptors on hepatocytes and are endocytosed, with remaining FFAs reesterified into intrahepatic triglycerides. Now, during the fasting phase, the liver becomes the primary source of circulating triglycerides. The intrahepatic triglycerides that accumulated in the fed state are exported by hepatocytes as VLDL, assembled with ApoB100 protein. FOXO1, active in the fasting state, stimulates ApoB100 expression. While adipocytes have low LPL activity during fasting due to low insulin, cardiomyocytes and skeletal myocytes express LPL, avidly digesting VLDL and providing FFAs to these muscle cell types for energy. This process generates intermediate density lipoprotein, or IDL. About half of the IDL returns to the liver for uptake and digestion. The other half undergoes further digestion by hepatic lipase, forming LDL. LDL is cholesterol-rich and very atherogenic. It's cleared by the LDL receptor, which recognizes only ApoB100, unlike the multiple EPE receptors. The liver is the major site of LDL uptake, clearing cholesterol for excretion as bile acids or storage as cholesterol esters.
Beyond the hormones and circulating lipids, what about the crucial transcription factors that truly drive these metabolic shifts at the cellular level? These are like the deepest genetic switches, aren't they?
They are indeed the master genetic switches. We need to highlight SREBP1C versus SREB2. SRBP2 resides in the endoplasmic reticulum membrane and is primarily a cholesterol sensor. In low cholesterol conditions, it's cleaved and translocates to the nucleus to increase transcription of genes involved in cholesterol synthesis and uptake. SREBP1C, on the other hand, is highly expressed in adipose tissue and liver. It stimulates genes involved in fatty acid and triglyceride synthesis and is powerfully activated by insulin and the ECA-P pathway. So one is about cholesterol, the other about fat synthesis. Then there are the PPARs, or peroxisome proliferator activated receptors. These are nuclear hormone receptors that heterodimerize with retinoid X receptors, or RXRs, and crucially bind to FFAs, acting as fat sensors. PPAR gamma, abundantly found in white adipose tissue, promotes triglyceride storage and adipocyte differentiation. It also stimulates adiponectin secretion, which remarkably improves insulin sensitivity. Mutations in PPAR gamma can lead to lipodystrophy, severe insulin resistance, and diabetes, while drugs like thiazolid indiones activate PPAR gamma, improving insulin sensitivity. PPAR alpha is found in the liver, muscle, and kidney, promoting FFA uptake and oxidation, thus acting as an antisteatotic molecule. Fibrates, a class of drugs, activate PPAR alpha, and PPAR delta, found in adipose and muscle, also promotes fatty acid oxidation, muscle stamina, and HDL production. Another family of lipid-sensing transcription factors are the LXRs, or liver X receptors. LXRs are also cholesterol sensors. In high cholesterol conditions, they upregulate ATP binding cassette, or ABC proteins, promoting cholesterol efflux from cells and its excretion. In the liver, LXRs also promote the conversion of cholesterol to bile acids for excretion or to cholesterol esters for storage, increasing SREBP1C expression to provide the necessary fatty acyl-CoAs.
So understanding all this incredible metabolic machinery, what does it tell us about why some people struggle with their weight and develop conditions like obesity? It seems so complex.
It tells us that energy storage is an incredibly complex balance determined by caloric intake versus expenditure. The vast majority of stored energy is fat, and individual variation in body fat has significant genetic components. Evidence from adopted children studies, identical versus fraternal twin studies, and the recent discovery of specific obesity genes all strongly support a significant genetic influence. Beyond genetics, fetal programming, where a mother's diet profoundly affects her offspring's adult weight and body composition, also plays a profound role. Low birth weights, for instance, correlate with increased risk for obesity, cardiovascular disease, and diabetes in adulthood, suggesting a thrifty metabolism can be programmed in utero to hoard calories. To measure adiposity, we often use the body mass index, or BMI, calculated as weight in kilograms divided by height in meters squared. A BMI over 25 is considered overweight, and over 30 is obese, both significant risk factors for insulin resistance, dyslipidemia, diabetes, and other conditions. It's also critical to differentiate between white adipose tissue depots, subcutaneous fat versus intra-abdominal or visceral fat. Visceral fat, though often smaller in volume, drains directly into the hepatic portal system, directly exposing the liver to its released FFAs. It's also more highly innervated and has a faster turnover rate, making it metabolically more active and problematic.
And this distinction between fat depots is why the classic apple versus pear body shapes matter so much for health, isn't it?
Exactly. Men tend to gain fat in the intra-abdominal depot, leading to an android or apple-shaped adiposity, while women tend to gain fat subcutaneously, particularly in the thighs and buttocks, resulting in a gynecoid or pear-shaped adiposity. Excess abdominal fat poses a significantly greater health risk for the pathologies we've discussed, due to its direct impact on liver metabolism and inflammation. That's why the waist-hip ratio, which measures waist circumference divided by hip circumference, can often be a better indicator of disease risk than BMI alone. Ratios greater than 0.95 in men or 0.85 in women linked to higher risk for diabetes and cardiovascular disease.
This raises a truly important question. With all these peripheral signals from fat and hormones, how does our brain, our central command center, know when to eat and when to stop? How do appetite and satiety work?
The hypothalamus is absolutely key to integrating these signals and regulating appetite and satiety. A simplified model involves leptin, an adipocyte-derived protein that signals the degree of adiposity and nutrition to the hypothalamus. In the arcuate nucleus of the hypothalamus, leptin acts on at least two neuron types. In one, it represses the production of neuropeptide Y, or NPY, a potent stimulator of food-seeking behavior and an inhibitor of energy expenditure. And it also represses a Goody-related peptide, AGRP, which is an antagonist of alpha-melanocyte-stimulating hormone, or alpha-MSH. In another neuron type, leptin stimulates the production of propromelanocortin, or POMC, products like alpha-MSH and cocaine-amphetamine-regulated transcript, or CART, both of which inhibit food intake. So, leptin simultaneously inhibits hunger signals and stimulates satiety signals. These second-order neuropeptides then transmit to the paraventricular or satiety nucleus and the lateral, or hunger, hypothalamic nucleus, coordinating feeding behavior and autonomic nervous system activity. Leptin is truly fascinating. Leptin-deficient mice and humans lacking this signal become morbidly obese, originally raising hopes for leptin therapy. However, here's the paradox. Diet-induced obesity often leads to leptin resistance. Obese individuals already have elevated endogenous leptin levels, but their brains simply don't respond to the signal, failing to recognize the satiety message. Leptin also plays a critical role in liporegulation, protecting peripheral tissues like the liver and muscle from accumulating too much lipid by directing excess caloric intake into adipose tissue for safe storage. Other critical regulators of energy balance include melanin-concentrating hormone, or MCH, which increases food-seeking and adipose tissue by antagonizing alpha-MSH's satiety effect. Its importance is underscored by the fact that its ablation by gene knockout actually results in leanness. Serotonin produces satiety after glucose ingestion. Gastrointestinal hormones like cholecystokinin, CCK, and GLP-1 also produce satiety. And then there's ghrelin, a potent orexigenic or appetite-stimulating peptide from the stomach. Its plasma levels rise dramatically in the 1 to 2 hours preceding normal meals and fall drastically afterward, appearing to stimulate food intake by reacting with its receptor in NPY expressing hypothalamic neurons, effectively ringing the dinner bell.
Now we come to the real-world implications. The point where this incredibly complex and finely tuned system goes awry, particularly with diabetes mellitus.
Right. Diabetes mellitus is a disease where insulin levels, or the responsiveness of tissues to insulin, or both, are insufficient to maintain normal plasma glucose levels. While diagnosed primarily by plasma glucose, diabetes often promotes significant imbalances in circulating lipids and lipoproteins, a condition known as dyslipidemia, which is a key contributor to its long-term complications. General symptoms include hyperglycemia, excessive urination, polyuria, excessive thirst, polydipsia, excessive hunger, polyphagia, muscle wasting, electrolyte depletion, and in type 1 diabetes, dangerous ketoacidosis. Diagnosis typically involves a fasting plasma glucose test. Normal is below 110 milli-GPL. Values between 110 and 126 milli-GDL indicate impaired glucose tolerance, and above 126 milli-GDL on 2 successive days confirms diabetes. An oral glucose tolerance test, where blood glucose is measured 2 hours after a glucose bolus, confirms diabetes if levels are above 200 milli-GDL. A non-fasting plasma glucose level greater than 200 milli-GDL with symptoms also indicates diabetes.
Let's clearly distinguish between type 1 and type 2 diabetes. Type 1 is distinct in its origin, an absolute requirement for outside intervention.
Type 1 diabetes mellitus, or T1DM, is characterized by the autoimmune destruction of pancreatic beta cells, often due to an inflammatory process called insulitis with mononuclear cell infiltration. This means virtually no pancreatic insulin is produced, and exogenous insulin is absolutely required to sustain life and prevent severe ketoacidosis. Its onset is typically in childhood, often between 10 and 14 years of age, though it can occur at any point in life. T1DM is often associated with specific islet cell autoantibodies and specific HLA types, particularly DR3 and DR4, indicating that strong autoimmune component.
And type 2 diabetes, which has reached epidemic proportions globally, is often so closely linked to obesity. What are the complex causes and typical progression here?
Type 2 diabetes mellitus, or T2DM, has complex causes, typically involving a significant degree of insulin resistance coupled with some degree of beta cell deficiency. The progression usually starts with insulin resistance, where the body's cells don't respond effectively to insulin. This initially leads to reactive hyperinsulinemia as the body tries to compensate by producing more and more insulin. Over time, however, this progresses to relative hypoinsulinemia, where the beta cells can no longer produce enough insulin to overcome the growing resistance, eventually leading to beta cell failure.
And drilling down on that, what are the 3 major underlying causes of obesity-induced insulin resistance in particular? This is where the whole system really breaks down.
The first major cause is a decreased ability of insulin to increase GLUT4-mediated glucose uptake, especially by skeletal muscle. This is often due to lipotoxicity. Excessive triglyceride accumulation in muscle cells, where byproducts like diacylglycerol and ceramides stimulate signaling pathways that actively antagonize insulin receptor and IRS protein signaling, essentially jamming the signal. The second cause is the decreased ability of insulin to repress hepatic glucose production in the liver. This insulin resistance in the liver is also often linked to lipotoxicity, leading to fatty liver or hepatic steatosis. Visceral adipose tissue, that fat around your organs, plays a significant role here, releasing inflammatory cytokines like TNF-alpha, which can antagonize insulin signaling. And its high turnover rate exposes the liver to high levels of FFAs, exacerbating hepatic lipotoxicity and further driving glucose production. The third major cause is the inability of insulin to repress hormone-sensitive lipase, HSL, or increase lipoprotein lipase, LPL, in adipose tissue. High HSL activity and low LPL activity are major contributors to the dyslipidemia associated with insulin resistance and diabetes. This leads to hypertriglyceridemia, with the liver producing large triglyceride-rich VLDL particles. When these VLDLs and their intermediate forms, IDLs, are efficiently digested, they give rise to small, dense LDL particles, which are very atherogenic. Furthermore, HDL takes on excess triglycerides, shortening its half-life and leading to lower levels of protective HDL.
And all these factors, insulin resistance, dyslipidemia, high blood pressure, often come together in what's referred to as the metabolic syndrome, creating a dangerous cascade of health risks.
They absolutely do. The metabolic syndrome is a constellation of risk factors that includes T2DM, obesity, insulin resistance, hypertension, and coronary artery disease. It represents an interconnected set of conditions that significantly increase an individual's risk for cardiovascular events and other chronic diseases, highlighting how intricately linked these metabolic pathways are to overall health. We've truly taken an exhaustive deep dive into the hormonal regulation of energy metabolism. It's clear that the body's sophisticated ability to switch between phases, using and storing different fuels is, well, it's a marvel of biological engineering. Understanding the intricate dance between insulin, glucagon, and catecholamines, and the roles of specialized cells and the detailed signaling pathways offers powerful insights into both the mechanisms of health and the origins of disease.
Absolutely. We've seen how specific transcription factors like SRBP1C, SRBP2, PPRs, and LXRs orchestrate these metabolic shifts at the genetic level, and how conditions like glucotoxicity or lipotoxicity can profoundly disrupt this delicate balance. It's a powerful reminder of just how interconnected our metabolic health is, affecting everything from your muscles' energy supply to your brain's hunger signals. This is truly mastering the information like the elite professionals in this field. So what does this all mean for you? Perhaps you'll consider the incredible, complex journey of a simple meal through your body with a new appreciation. Or maybe you'll ponder the profound long-term implications of even slight imbalances in this finely tuned system. The challenge of maintaining energy homeostasis is constant, and understanding it is truly a step towards mastering your own well-being.
Indeed, consider this provocative thought. Given the immense genetic influence and fetal programming on our metabolism, how much control do we truly have over our metabolic destiny? And how might future personalized medicine leverage these deep insights to tailor interventions more effectively, moving beyond a one-size-fits-all approach?
Join us next time for another deep dive, where we'll turn another stack of sources into clear, actionable, and masterable knowledge. Until then, keep learning.